Can you see my slide?
Yes, it looks like
Great.
them. Folks are
Hello,
still seeing.
everyone. I'm Kyoji Tsuchikama. I'm associate professor at the Texas Therapeutics Institute, the IMM, or Institute of Molecular Medicine here at UT Health Houston. Thank you for giving me a great opportunity to share our recent academic research achievements, and I'm really pleased to show what actually we've done in the past years, and then how we translated our basic scientific discoveries into real products that has a commercialization potential. So the next speaker, Dr. Michael Torres, who's the CEO of a newly formed company, Crossbridge Bio, will take the part. So today I'm going to talk about a little bit of some basics of the antibody-drug conjugate, and then show some summary of or highlights of our key technologies. So here's my disclosure. I'm a co-founder, a member of the director board, and a scientific advisory board of the company Crossbridge Bio, and I also hold equity in this company. So my lab is located at TMC3, as shown in these pictures. It's a very nice, beautiful, and futuristic research center with many academic and industry labs living together. So my lab is on the second floor of this TMC3 building, and the Crossbridge Bio is on the third floor. So we are very well connected with almost zero distance. So this is a very unique and productive place to form academia industry collaboration. So this is where we're working. And I think most of the audience or people here know about the ADCs, or antibody-drug conjugates, but this is the graphical summary of the technical features of the ADC molecule. So ADCs consist of three major parts: antibody, the payload, and the linker. And also how the linker and payload is connected is also a crucial element, making ADC of interest, good or bad. So my lab is a medicinal chemistry lab working on the payload and linker and conjugation chemistries mainly. And the host, Dr. Zichen An, is an expert of the antibody engineering, discovery and engineering. So we have formed a very productive collaboration with his lab, and then we have generated novel ADCs using our unique and very efficient payload, linker, and conjugation chemistry platforms. So by using ADCs, you can achieve much higher cancer cell killing potency without sacrificing the therapeutic window. And you can also significantly extend the circulation life of the drug injected, thanks to the antibody's long circulation life. So as you know, the ADC field has grown significantly, and now we have 13, or it could be already 14, including some of the newcomer ADCs approved in recent years. And also we have so many ADCs in clinical trials. That said, the development of ADCs is not an easy task. As you may know, there are so many new companies, including Crossbridge, and Crossbridge is an exception. I'm really happy with that. But so many new companies actually discontinued many development projects, and some companies have already, closed down their business. So that's because if the molecule or payload is conjugated with suboptimal linkers, with suboptimal stability or payload release properties, it messes up the overall ADC safety and efficacy. So this linker chemistry has to be carefully designed and refined, not to see it unsuccessful products in the end. So with that background in mind, we have developed very unique, simple, but powerful solutions to maximize both stability, tolerability, as well as other um, biophysical properties. So one example is this tripeptide linker called glutamic acid glycine citrulline. I'm going to talk about the details later, but this significantly improves the ADC stability without sacrificing the ADC's anti-cancer potency. Another achievement is a homogeneous site-specific conjugation platform, as shown in the bottom figure. Most ADCs have been developed using random conjugation methods, giving a mixture of varying antibody drug conjugates with varying antibody to drug ratios. So this is a very heterogeneous mixture. But by using our site-specific precision conjugation method, you can install your payloads onto the specific targeted position with desired antibody to drug ratios. In addition, we can also combine two different payloads, payload 1 and 2, at desired ratios, and then create dual payload or dual drug ADCs. So these two key technologies have been given to the Crossbridge Bio with exclusive patent license. So now they are actively developing truly innovative and therapeutically potent ADCs. So with Dr. Michael Torres will explain their ongoing efforts on their end, but I'm going to talk about how we actually discovered such novel linkers. So the graphical uh, uh, this is the chemical structure of the industry standard linker called valine citrulline or val-cit linker. So this linker is designed to be cleaved at this peptide bond in response to um, proteases called cathepsin. So that is highly expressed in many cancer cells. So once this bond is cleaved, the conjugated payload molecule is released without any trace. So this has been a gold standard in the field, but previous studies have also shown that this linker is not perfectly stable, and it can get cleaved by rodent plasma, and it can be also cleaved by other undesired proteases, such as neutrophil proteases. So these two issues complicate not only preclinical evaluation using mouse models, but also clinical applications without seeing toxicity issues. And indeed, valine citrulline-based ADCs have shown many toxicity issues, such as ADCETRIS 6, 20, 29. They suffer from some level of neutropenia and other associated side effects. So the improvement of this linker system was a critical issue at the point. And after thorough screening of the linker structure, we finally found a very simple modification that resolves almost all issues. That is the glutamic acid plus glycine structure. So original structure didn't have the glutamic acid, and or they also had valine but not glycine. So we found these two specific modifications significantly improve stability as well as the hydrophilicity of the entire molecule, and of course potency and safety profile. So this is just a summary of the early stage screening of the linker surrogates. So I'm not going to talk about the details here, but what we found in this experiment is that by introducing glutamic acid and glycine, you can completely protect the linker structure against the degradation mediated by neutrophil elastase. And take a look at this green curve, which is, oh, sorry, not this one, the purple one, B-sit or val-sit. This is the gold standard linker. So this does get cleaved. Oh, sorry. This does get cleaved by human neutrophil elastase. And mouse plasma stability is also improved by our modifications. And of course, there's no degradation in plasma. That holds true for the valine citrulline linker, but as I showed in the first graph, it is unstable against neutrophil elastase and other similar proteases. So our linker was the most stable linker. And as additional bonus, we could improve the hydrophilicity significantly. So this is HIC or HIC chromatogram. Uh, as you can see here, our EEG-CIT-based ADC showed much earlier retention time compared to the traditional valine citrulline-based conjugate. So the earlier retention time means it's more hydrophilic. So that means it has less tendency, the aggregation tendency, and it's potentially advantageous to minimize the clearance from the body due to reduced aggregation tendency. And we also verified the improved safety or tolerability. So we created MMAE ADCs at DAO4 using our linker, and we also tested ADC approved in HER2 and Kadcyla. As you can see here, we didn't see any trace of liver-associated damages based on the elevation of related enzymes such as ALT and AST. And while Enhertu and Kadcyla showed some level of cell damages in the in the slides, we didn't see there's almost no damages to the liver tissue here. So this is one of the demonstrations of the improved safety thanks to our EEG-CIT linker. So Michael will explain a little bit more details how this linker is stable and how this is safe compared to other competitors' technologies. And as I explained earlier, we have also developed dual payload conjugation technologies. So this is the short summary of what we can do using the dual conjugation platform. Using that, we can achieve we can have two MOAs or mode of mechanisms or actions within the same single molecule, and it can potentially broaden the scope of target on tumor cells, or you can effectively kill subpopulations with elevated resistance to either payload 1 or 2. And thanks to its unified single molecule platform, we can expect a simpler PK compared to one-to-one cocktail of two different ADCs. But of course, there are a couple of challenges. One is we can incorporate similar numbers of payloads, payload 1 and 2. So those two payloads have to be carefully chosen based on expected similar potencies. Otherwise, one can work very well, the other one may not work well. So the choice of two payloads is very important and very difficult to identify. Also, there are a couple of risks, such as additive or synergistic toxicities, and also both payloads are delivered and released at the same time. So sequential or delayed delivery may not be easy to achieve. And also, we have to establish a very efficient production. Otherwise, if it's too complicated to make, it's not going to be practical drug design. So after our thorough screening, we have established very efficient conjugation scheme as shown here. So this is our lab's achievement, or the best effort. But surprisingly, Crossbridge has already developed a much more efficient production scheme. So I'm really let's I would like to leave that kind of details to Dr. Mako. But what the point is, by introducing this heterofunctional branch linkers using microbial transglutaminase, which mediates site-specific transpeptidation at glutamine 295 position, you can get heterofunctional branch linker antibody intermediate. And you can use this intermediate for a variety of payload combinations by mixing and matching payload 1 and payload 2 modules. In this case, we have click reaction handle MMA module and another click reaction handle and MMAF modules as an example. So by adding these two one by one, you can get the final dual drug conjugate with relatively high yield. And this process is pretty much scalable. And even with our very basic setup, we can achieve highly homogeneous conjugation with just single peak at the intermediate stage, as well as almost single peak at the final stage. The other two are just fragments by mass spec and ionization. So this is a single product. And also, our conjugate platform gives a high monomeric product, so we don't have to worry about aggregation issues. So this is our recent data using the TROP2 antibody. So we constructed MMAE, MMAF dual payload conjugates using our own unique TROP2 antibody. And we treated human xenograft mouse models, or TNBC, triple negative breast cancer tumor model. So we injected our dual drug ADC at 3 mg per kg just once as a single dose, and then monitored the tumor growth. And as you can see here, just one single dose was good enough to completely suppress the tumor growth. And as you can see here, one FDA-approved TROP2 ADC, named sacituzumab govitecan, was not potent enough to suppress the tumor growth, as you can see in this green color. And another interesting point is that we also made the same dual payload ADCs, not using our own TROP2 map, but these sacituzumab, which is again a part of the FDA-approved ADCs. So this SASI dual payload also showed the comparable, very potent anti-tumor effect. So that means our dual payload platform is not really dependent on what antibodies are used. So it's a very universal linker platform that can be used in any antibody scaffold. Okay, so we have just less than 5 minutes. So let me show our very recent data to expand our portfolio beyond the traditional ADCs. So as you may know, uh, linker conjugation technologies can be used not only for cytotoxic payloads, but also for other payload classes. One example is immune stimulating antibody conjugates, or ISACs. So instead of installing cytotoxic payloads, you can install immune stimulating payloads. So that is another new modality many pharmaceutical companies are working on. So we actually further evolved that Isaac by introducing additional payloads to the standard Isaac structure. So here we have four additional undisclosed payloads in addition to the traditional immune stimulating payloads here. So we named it next gen Isaac. A traditional Isaac molecule does not have these additional yellow spots. And we treated a human HER2 expressing syngeneic tumor models by Isaac or our next gen Isaac. And we also tested standard ADC on trastuzumab exatecan DAR4 conjugate. And here's the result. So ADC, uh, because this model is very aggressive and hard to treat with traditional ADCs, it showed exatecan ADC showed a very modest effect. And Isaac worked slightly better than exatecan ADCs, but it was not satisfying. Our next gen Isaac showed improved efficacy. And when we combined this next gen Isaac and exatecan ADC as a combination therapy, well, there's no significant difference in tumor volume, but the probability of survival was significantly enhanced. So we could observe 5 out of 8 tumor-free mice in this combination group. So we further tested its effect by rechallenging the survivor mice with the same tumor line, but without HER2 human HER2 expressions. So this is the wild type CT26 model. So this way we can test if the survivor mice can reject the implantation of the wild type tumors without HER2 related immunity. So this is the result. So after the second challenge of CT26 wild type tumors, we didn't see the tumor regrowth. That means these survivor mice developed robust memory, anti-tumor memory formation against this cell line, not against just HER2, but other tumor-associated antigens derived from this tumor line. And after confirming no tumor growth, we injected 41 tumor, which has completely different genetic background. And that tumor grew pretty quickly. So this means the anti-tumor memory was specific for this cell line, not for any other random tumor lines. So this is one demonstration of our next-gen Isaac platform, and hope we can disclose more technical details once this manuscript is submitted. Okay, so I have many things I want to share with everyone, but the one message I can deliver is this dual payload conjugation platform is unlimited. It's not limited to traditional ADC or even ISAC. We can change the payload classes. So to make uh, mo completely different modalities. One demonstration is payload, cytotoxic payload, radionuclide combinations, or radioactive ADC platform. So this has been published, and please take a look at this manuscript if you're interested. And also, we're currently working on antibody oligonucleotide conjugates. So this is a very new approach that many pharmaceutical companies are really excited about. So, in collaboration with Dr. and Dr. Wenbo Lee at the TMC3, we have already established very efficient oligonucleotide conjugation method. So we can achieve DAR4 conjugation without any undesired uh, cytoreactions. And so now, I have to say thank you to the Gulf Coast Consortium. To our another project has been funded by GCC for a very similar project, but siRNA AOCs for prostate cancer. So we can also conjugate siRNA instead of single-strand oligonucleotide. So it's still in its infancy, and we still need to improve the conjugation efficiency. But by optimizing the conditions, we can achieve relatively homogeneous conjugation of double-stranded siRNA conjugation, as shown in this gel band. So we are trying to improve this process, and then hopefully we can share more information in the future seminar. Okay, so here's a summary. So there's no limitation in this dual drug conjugation platform. We can expand our portfolio based on our biological hypothesis or new approaches. And here's my acknowledgements. I would like to thank all of my collaborators, my lab members, collaborators, especially Dr. Zijian An, and other collaborators and funding sources. Thank you.
Thank you, Kyoji. And now we have Marco to follow up the commercialization of the ADC technology at Crossbridge.
Yep. Thank you, Jishang, and thank you, Kyoji, and thank you to the audience for joining this talk. I think we'll get to it, throughout. But what I'd like to do again is show you how we've kind of taken what's seemingly very promising technology, and it continues to play itself out that way, and build a company right here in Houston. And so I'm hoping that others, can take the learnings of how you basically build a best-in-class company, right here in our ecosystem, or anywhere else for that matter. So and I'll layer in my background as well. So just again as the sort of high level overview, and I'll share more details. But this we started this company in 2023 again with Kyoji, and Jishang's also a co-founder as well. And really with the mission to translate this promising technology into clinical practice and hopefully transformational medicine. So we do have a lead asset that's called CBB-120, which is a dual payload TROP2 ADC. And again, I'll give you more details on how we arrived as a company to the lead asset that we have, and then give a view to where we are, which again, we're targeting an IND filing. So I'm personally excited to, again, having this technology been in the tech transfer office and in the lab setting not too long ago, that we're on track to be, have filing an IND this year, and be in the clinic shortly thereafter. And having done that, or having gotten there, was really also important and centered on this Texas ecosystem. So as you see, we've actually raised about 15 million from CPRIT. So CPRIT's a great resource for those of us here and those of us who want to build in Texas. And so I'll give more details, but we actually started with a $3 million seed grant we hit all our milestones very quickly and applied for a $15 million follow-on grant, which was awarded. So, me and the company are two-time CPRIT awardees back to back. So I'm also very proud of that. And then we were able to leverage, investors in the local ecosystem and around to start moving the company forward. We raised 10 million in a dilutive financing, and then actually prior to that, I did about a 1.5 million pre-seed friends and family financing, which now again, as I mentioned, we're on track to be in the clinic. So really, I mean, there's no real big secret to how you translate, technologies and get it towards a clinic. I mean, it starts with fantastic team. It starts with good technology, and then you need experts in their various, areas of focus or subject matter experts in various areas to really move a company forward in technology. So a few folks that I want to highlight is my CSO, is this guy named Dan Pereira. Dan's been in the industry for a very long time, has over 5 FDA approvals under his belt, and actually, at one company, they filed an IND for an ADC that is one of the ones that are approved and on the market. So it's that level of expertise that's really required to go from an early stage proof of concept data set, high quality publications, and decide like how to build an ADC that then can be manufactured, that can be tested in safety studies, and go through the drug development path. So Dan's very important to that. In our case, we also have an expert in manufacturing named Morris Rosenberg. He's been developing biologics and ADCs for over 30 years and was on the commercial launch team on the manufacturing side of many of the ADCs that are on the market today, including Trodelvy and Adcetris. And then I mentioned our scientific co-founders that we started the company here. And then again, various other folks, including Alice, who's a clinical trialist, designer, expert. And again, being here in the Houston ecosystem, we're able to take advantage of, world-class clinicians and advisors out of MD Anderson as well. And that has been really useful as we thought about what does our phase 1 look like, and the types of patients that we're going after. And so again, the take home is you really need to have people who know what they're doing. In my case, I'm a scientist, PhD from UT Southwestern. I started a company out of my postdoc called Recode Therapeutics that raised 400 million, and we had the company's still around, 2 assets in the clinic. We had, some major investors, including the venture arms of Pfizer, Sanofi, Amgen, and Bayer. So for me, I can leverage all of those relationships to, get in front of investors, and that's what you need to do to, translate. You need money, as we all know. Okay, so Kyoji touched on this, but so I'll just give you like the high level company view of like what we're trying to do. Again, when it comes to ADCs, they're very promising drugs, but safety, efficacy, and addressing resistance are the 3 main things that we're still trying to solve for. And so, Kyoji gave a view of the technology, but just to say it our way, our solution relies on site-specific stable conjugation of branch linkers that then with the antibody we can be Fc silent or have N-glycan intact antibodies, and that can make a difference in the safety profile by way of interacting with FcR gamma receptors, and that's important in it's essentially non-specific uptake of your ADCs, and you don't want that because you don't want the payloads going where they shouldn't, right? And then I think Kyoji gave a good view of the EG-CIT linker and why we're particularly excited about that in terms of its sensitivity to cathepsin B, a known cleavage mechanism, and also the resistance to the normal tissue proteases. And again, as we've thought about the problems that we want to solve, to be able to have flexibility between payloads that's and the DAR and how many you put that's very desirable because the take home is, we know that combinations work, but combinations can be toxic, especially if delivered systemically. So if you could put them in on ADC where they can better direct towards the tumor, you could potentially unlock biology within a tumor cell that is harder to overcome. And in our case, our lead payload combination, and we'll share more about that later, is a topoisomerase by ATR inhibitor. So an ATR is a DNA damage checkpoint or repair protein. And so if you block that, topoisomerases cause damage. You essentially exacerbate, or in our case, we show really nice synergy between the two payload pairing. And so synergy means better potency, and as long as it doesn't fall on tox, that's a very highly desired potential drug candidate that you want to go after. Kyoji touched on some of this already, but just to give again the company view, this is looking at the hydrophobicity of the ADC. And in this case, we have sexatuzumab, so that's actually the antibody that we use in our ADC. We have a mutation that makes the DAR4 facilitates the DAR4 plus 4, and that then makes our EG-CIT dual payload ADC, and so that's here in red. So these are the naked antibodies. How fast they elute relates to how hydrophobic they are, and so the quicker they elute, the more hydrophilic. And in this case, what we did is we took two drugs that are on the market. This is Trodelvy and Datroway, two ADCs that are approved. We swapped out the antibody to be the same antibody across each one of these ADCs, and then we put their linker payload together, which is this is Trodelvy right here, and this is Datroway, their linker and their payload. And so now we can compare apples to apples across the hydrophilicity of these ADCs. And as you can see, our ADC is much more hydrophilic, which again is a desired property. This is looking now at the cleavage that I mentioned. And so in this case, we took different linkers that are used in various drugs, and we're looking at the cathepsin B kinetics. So here you can see that this is us in red, very fast. Everybody has similar kinetics, but the extent of cleavage can vary between different linkers. And so you want to be very sensitive and have complete cleavage. And that's what we have with our EG-CIT linker. And then importantly, this is looking at neutrophil elastase and cathepsin G, so normal tissue proteases. And again, we're looking at various linkers. And here we've listed some of the drugs that use these linkers. And as you can appreciate, our EG-CIT linker is very stable to these proteases, whereas Valala, Valcit are very sensitive to neutrophil elastase, and Kyoji, already mentioned about neutropenia, and then GGFG is very sensitive to cathepsin G. And so again, as you can appreciate, especially here, if you look in this small table, some of these listed assets are actually some of the new dual payload, like others trying to do dual payload. And as you can appreciate, they're using different linkers and ones that are off the shelf and available to build their ADC. And as you can appreciate, there's different sensitivities to these with these linkers. They have their own liabilities, or we'd have a lot more approved ADCs that use these linkers. And then if you try to sort of match up kinetics and stoichiometry to get two drugs in one cell at the same time, if they have different sensitivities to proteases, that we feel that's a very suboptimal design. And so in our case, it's the same linker, payloads are released at the same time in the cell, and we can show that biological effect, which we'll get to in a bit. So again, very high marks for the linker platform. And then to walk through our asset today, our lead asset, again, this is mentioned, we have sexatuzumab. So this is also a validated antibody used in several, at least one commercial drug in the US. There's other folks working with this antibody, so there's a lot of data around it. We talked about the DAR4 plus 4 using the branch linker and the microbial transglutaminase, as Kyoji mentioned. And then we have it FC silent, which again is important for nonspecific uptake. And then I'll actually walk through how we got to the payload pairing that we did. and how we're utilizing this to move it forward. And it's a footnote here, but this is again highly manufacturable. We've now almost done with GMP manufacturing, and to say it this way, with our CDMO, we started this process last year in May, and we haven't had to shift the timeline at all. And we're going to have multi-grams of GMP material for our phase one. And so again, really speaks to the chemistry, the design of the ADC, and how manufacturable it is. So, a lot of folks ask us, why TROP2, why this particular asset? So we like to give a view to this. And so again, as I mentioned, there are clinical benchmarks for TROP2 and various preclinical ones that we can benchmark. The two drugs that are approved are for subsets of breast and lung, and they're both billion-dollar franchises, and TROP2 is widely expressed across many different tumor types. And so again, that makes it a highly attractive target. And right now, many of the ADCs, that are approved, and the two tropes that are approved use topoisomerase inhibitors. And so, although we have one, a topo on our side, we also have this ATR, which again, we'll show you, we're actually can address resistance with this combination, but it leverages a payload class that we know is effective in patients, which is also, very important when you're thinking about how to de-risk a program. And so, yes, this is again, the rationale for CBB and the potential, right? So it's CBB 120, so it's a 1-2 punch on the DNA damage pathway. We have really good potency. You'll see the safety data later, and it is expected to be synergistic because of the mechanisms, and we are showing that in efficacy models, and you'll see some really cool PD data as well to show that we are actually engaging and inhibiting ATR, which is actually pretty cool because ATR is a kinase, and so there's been big questions of can you get enough drug within a tumor cell that's not a cytotoxic to have a biological effect, and we have data to show that. And again, as I mentioned, we have the opportunity to be effective in obviously topoisomerase naive patients, but then potentially in refractory patients as well, which is also very, very important when you're building a drug. So again, just to walk through the asset a bit, I won't spend too much time. These are looking at the binding of the sacituzumab to human TROP2 and cyno TROP2. Again, single digit KD, so single digit nanomolar affinity. This is highly desired and what you'd expect for sacituzumab. So again, we sort of check that box. In this case, this is the FC gamma binding that I mentioned. And so this is sacituzumab and Dato-A or Dato-DXd, the approved, these are the benchmarks. And these are ADCs in single and dual payload formats. So as you can see with the gamma 2 and 3 receptors, we don't bind, but we do bind to FcRn, which is important for half-life. And so again, we have a normal half-life for an antibody and ADC, which is very, very highly desired characteristic. And then so in this case, just to give a view to, as I mentioned, how we got to the payload pairing that we did. Again, we thought, okay, let's look at topoisomerase, and then what are drugs that would be predicted to be synergistic with that. So one immediately thinks about DNA damage inhibitors. And so we actually screened several different DNA damage inhibitors. So PARPs, CHEK1, WEE1, PARG. So again, these are known DDR inhibitors. And it was actually in our ATR screen where we saw, we tested different ATRs, and it's actually this one we call ATR2 here was where we saw synergy. And so synergy is defined where, you have cell, in this case, cell killing that is, greater than each individual drug. And so, 1 + 1 is, more greater than 2 is essentially like the take-home version of that. So you can see that there. This is synergistic. And then I'll show you more data about that as well. So again, now with the ADC. So that was free payload screening. This is now the ADC. So in purple, this is the ATR. This is the ADC by itself, ATR by itself. In black is the topoisomerase by itself at DAR 4. We did a lot of DAR screening that I won't cover here. And then in green is Dato-DXd, the clinical benchmark. And this is our dual payload ADC at the DAR 4 plus 4. And so as you can appreciate, this is very highly synergistic where, you can see the IC50s of each individual curve, and then it's significantly more in the dual payload format. And we have multiple cancer types and multiple cell lines where we see this synergistic effect. And then just to kind of go back to how you think about your pipeline and how we thought about it, again, what we did is we took that lead ATR and topo payloads and we did a free payload screen. And in this case, this is each column is a different cancer type. And we actually did a 10 by 10 matrix of, TOP and ATR. And in this case, this allows, and we did the kill curves for all of those combinations. And what this allows us to do is approximate DAR of each payload as well, because if you have low of payload 1 and high of payload 2, you could think low DAR of one, high DAR of the other. And in this case, what I'm showing you is the one-to-one curve, and each cell is a different cell line if it's colored. So again, we saw that there were multiple tumor types where the TOP by ATR was synergistic. And so again, this helps us think about other targets that we'd go after. Say, for instance, if this is like colorectal cancer, you could think, what are the good targets for colorectal cancer, where we know that we're seeing, synergy within these tumor types. And then again, we also saw additivity, and then there were several cancer types where it was just very dominated by the topoisomerase. So again, this helps us think about our pipeline. In this case, these are now in vivo models, xenograft models. So this is a endometrial line that overexpresses TROP2, and this is the full graph here, but I'll zoom in and walk through each one of the curves. So in this case, we have the ATR ADC with our linker. Again, single payload. We have Dato-A, the benchmark here in green. And then we have our single payload ADC with the topoisomerase with our linker, and then the dual payload ADC with the TOP by ATR. And then we did a single dose of 6 mg per kg. And then because we wanted to see CRs, we did a second dose, but we can show that we can get CRs with a single dose at 6. And I can share more about the data later. But what you can appreciate here, again, the second dose of Dato-A was not effective at all in this model. And then with the second dose between our single and dual, we actually had about a month of CR. So we're actually surprised by how potent just our linker tech on the topoisomerase ADC, how effective it was compared to Dato-A. But then over time, those tumors started growing back in the mice. So we ended up with about a 50% CR rate. So complete regressions, about half the mice. Again, we think this is like what you see in patients, single payloads, patients generate resistance. But then with the dual payload, it was very effective, out to 4 months was 100% CR. And again, this is why we're excited that, this could potentially even subvert resistance if it's, potent and safe enough. And then, if we can still be active in the resistance setting, that's a huge win. And I'll show data, I think, in the next slide as well. So this is endometrial line. This is a lung cancer line where we see that always we beat the benchmark, but that the the dual payload, is more effective than the single payload. And, even in the case where, the lines don't separate, our view is, again, having that, dual 2 payloads, as long as it's safe enough, would be, what clinicians would prefer over a single payload. And again, down here on the right just gives you a view of, like, it's a dose titration where we also did PK as well. So that also helps with calculating your therapeutic index. And so really in most of our models, we see tumor regression between 2 and 4 mg per kg, which is, pretty good for efficacy that you'd want to see. Again, Almost done with my slides here, but in this case, now we're looking at resistance models. So this cell line is known to be resistant to Enhertu, which is an approved HER2 ADC. So if you look down here in the bottom left, this is at 3 mg per kg with everything. So Enhertu isn't effective compared to vehicle and in like purple, but our ADC at 3 mg per kg has some activity, albeit it's not super great, but it's a low dose too. So at 6 mg per kg, you see much more activity with our ADC versus in HER2. And then at 10 mg per kg, in HER2 actually now starts to have some activity, but gives back and tumors start regrowing, whereas we have really nice regression and tumor leveling, through an extended period with our ADC. And over here on the right, these are models that are resistant to Datroway, so the approved Trop-2 ADC. So in this case, this is a cell line that's known to express efflux pumps that can pump out DXd. So that's what you see here. In this case, this colon cancer line, Datroway is not active at all, whereas our dual payload has really nice activity in this model. So it's not a substrate of this efflux pump. And then here is a triple negative breast cancer line that also overexpresses Trop-2, where Datroway is really not effective at all. And we have activity, albeit, this is, very clear, a very challenging model. But overall, we are very happy with the amount of activity that we're seeing. Um, I think it's the last two slides. In this case, this is PD data. So again, as I mentioned, we have folks asking, can you actually engage ATR? So here on the top right is a blot looking at the topo ADC by itself at two different dose levels. So we'll focus here. And so as you can see, topoisomerase can cause DNA damage. So you see upregulation of ATR, phospho-ATR, phospho-CHEK1. So these are DNA damage and repair markers. And then gamma H2AX is a marker of DNA damage. And then with the dual payload ADC, you can see that ATR is inhibited, which you'd expect. And the downstream marker of that, phospho-CHEK1, is inhibited. And then you see increased gamma H2AX. And then in terms of an orthogonal assay, this is now cell cycle. So if you have the TOP ATR or the TOP ADC, you're going to get cell cycle arrest. And this is what you see. Cells accumulate in S phase. If you block that arrest, cells will progress through the cell cycle into G2M, which is what you see. But that DNA is not repaired. It's, and they can get mitotic catastrophe and, DNA damage and cause apoptosis. So a really nice data set. And we actually have in vivo data that correlates with this, what we see in vitro as well. So really excited about that. And then just a word on safety. Again, this is really important. We did a very comprehensive 28-day study in non-human primates, and we dosed 10, 30, and 60 mg per kg Q3 weekly. So it's a repeat dose. We did full histopath, ClinChem, ClinPath, essentially the works. And the take-home message are, again, we had very good half-life, very stable. So that's indicated here where we at 30 mg per kg, and it's same thing for all the dose levels. This is the total antibody, total ADC, and curves, and they're right on top of each other, which means it's very stable. And then these down here in these purple lines are each individual payload detected by mass spec. And I mean, it's basically undetected, very low levels, showing that it is very stable. And as we talked about the off-target effects, neutropenia is a known adverse event for ADC. So even at 10, 30, and 60 mg per kg, this is looking at neutropenia. This is the upper limit of normal, lower limit of normal. This is the pre-dose in the NHPs, and there's no neutropenia here. It would fall below the black line, and actually 60 mg per kg is the green line. So again, very, very promising data. So if you take the PK exposure of in the monkey at 30 mg per kg, where at 60 we saw some pronounced skin effects, but at 30 very mild. So we know our HNSTD is between 30 and 60. You can calculate a TI by exposure, and we have a triple digit TI, which is a very high TI for an ADC, especially a Trop-2 ADC. So with that, I think this is why we're really excited to continue to move this platform forward. We've gotten, we've got money to get it into the clinic. We're about to do GLP talks in April of this year. GMP manufacturing is done in July, and then we'll be filing our IND, our first IND later this year. And so I think it's a really cool story of how you put all of these pieces together to create something that hopefully is transformational for patients. So thank you.
Thank you, Marco. Also, Kyoji. So I think we're open for questions, right?
Yes.
Mhm.
Yep. So I think I see a question right here. So I can answer that. Uh, yeah. So again, we have looked at this in multiple ways, and I can't disclose all of the search. Someone's asking about microbial transglutaminase and Kyoji. You may have a view as well working on this, but I can tell you that we definitely know Araris and Taiho, I think is what you're referring to, and they picked up some patents from Innate Pharma so two high level comments I can say. One is, I believe those patents are going to be expiring in 2033. So that's generally for everybody in terms of FTO. But in this case, our understanding of that technology and those patents, that it's very specific to how they're doing that, and we do it differently. And so beyond that, I can't really comment on others, but that's how I can answer that question today.
Yeah, when we developed the MTGase-mediated conjugation, actually there was a very, a seminal work by not this company, but one of the co-founders, Dr. Roger Shibley, who originally discovered MTG, you know, mediated conjugation using a linear linker. And in their papers, they well, not clearly mentioned, but they emphasized that only simple small linker can be conjugated. And if the linker becomes big, it's very difficult to achieve the homogeneous conjugation using this technology. But we doubted it. And then we improved the conditions and improved that. Yeah, with just optimal optimization, we can also engage dual conjugation, enabling branch linkers. So that's how we originally discovered our way to diversify the linker structures, and that was not covered by the original patent. So that's why we could file a new patent. After that, well, I don't know what actually happened, but they wanted to cover that dual conjugation technology as well, but it was after our IP filing. So they, tried to, incorporate very specific modification to the linker structure so it can stand out as new feature that is not covered by our filing. So they kind of came up with some detour to establish their own structure. So that means they have to always incorporate that specific Linux features, even if that is not really important or necessary. So that's where we are and as of today.
Cool. And those are all our opinions and not legal binding
Yeah, yeah.
words here. Very
Hopefully
nice.
we can evolve ourselves together without any fights or
Okay, so
something.
there's Kyoji, if I say there's another question, it said, what is the threshold percentage of a monomer to be considered a homogeneous ADC?
In the industry, I think 5% is sort of a target. If it's less than 5%, I think it's okay, at least, in the initial stage of the product development. But as an academic researcher, I want to try to, keep it less than 1% or up to 2%. That's our, standard. So this way, we're gonna have worry free, you know, uh, easy development in the once the technology is translated, right? But yeah, in the industry, it's around 5%.
And so I just want to add
Oh,
that
sorry, is it 5% oligomers or yeah,
1% oligomers.
aggregates?
So you
Yeah,
need to have
and
a,
more than 95%
yeah,
is
95% of
yeah,
monomer.
monomer. Yeah,
Yes.
sorry,
Mm,
it was an
mm,
opposite.
yeah. So actually, there's a either during the target validation. Let's say you have a target and that you want to validate if it's an ADC target. So you could actually can tolerate some so less pure, let's say 80%, let's say. And if you can demonstrate proof of concept in vitro, in vivo, then you can invest more into the ADC, the homogeneity of your ADC. So
Right.
depending on which stages of if it's a research stage, you can tolerate less purity than like when Kyoji had over the development to Marco for drug development. That's a much more stringent bar to follow.
Mhm.
Mm.
Yeah, maybe one thing I'd add to that point too is as Kyoji showed in his slides, in research in bench scale, they were hitting 75% yield from antibody to dual payload ADC, which, to be frank, is actually very good that's a good number for a single payload ADC, and that's very good for a dual payload ADC. And as we've scaled up with CDMOs, I mean, we're well over the 80% yield as well. And so I think again that there's a couple of things that you can learn from that. One is a good, choose your CDMO wisely is maybe one one thing to think about, and then the other is, when you can start off with really good technology, right? Like it just makes things so much easier. And to say it this way, I think especially as we're fundraising and we're working on deals and so forth and so on, pharma is quite impressed that, again, as I mentioned at the beginning, that 2.5 years ago this tech was in the tech transfer office, and we're going to be hopefully in the clinic, on track to be in the clinic this year, and certainly done with GMP manufacturing and IND filing. And so you can put all these things together again when you start with good technology. And so I think that's, to anybody who's, an academic in the audience thinking about like, how do I like, get my stuff out and as quick as possible. Obviously, there's a lot of other things, licensing, tech transfer, IP, you could talk through all of that. But I think again, the faster your technology idea, what have you, looks like a real drug, if you will. And so in this case, we're going to be IND enabling, so forth and so on. That's very attractive to pharma, very attractive to VCs, because then they can feel like, okay, this can be a real drug. Because when it's a paper, an academic paper, sometimes it's, this is really great science, but can it be a real drug? And so the more it takes development, to really answer that question. So I think, again, this is why it's important to try to think about these things early on when you're, developing tech.
So while waiting for others may have other questions, but Marco, let me ask you a question. So CrossBridge is in process of making the GMP lot that will support uh, preclinical tox, GMP tox, as well as phase one. Are you going to produce enough material potentially support the phase two or just phase one?
Yeah, so right now we've completed our, basically our tox batch for our GLP tox, which was a 200-liter run of our antibody and then the ADC. And then yes, for our phase one, we're essentially looking at about 50 patients worth of material that we'll do, and then yeah, we'll have to do a resupply as we expand further. So I think that's essentially what we're targeting right now. Which it's always a balance. Like if I had just a lot of money, I think we could really really scale up. And so I think, as of today, unless we're doing this with a partner, we'll have to have a resupply between then.
Yeah, so I think my question really is a regulatory question. So what's the latest FDA guideline for bridging different lots? For example, in the early days of antibody, when I was working antibody at Merck, so you need actually, if you produce the material at a different lots, you have to bridge it, make sure the two lots are identical.
Yeah, I mean, you'll always do your potency, your release assays, right? So, you'll set your specs
Okay.
for
Yeah,
that. And with
you
the
have to do
specs,
that.
yeah.
Yes. Okay. You still have to, every batch, every lot, you have to bridge it. Okay,
Yeah.
good.
As long
Okay.
as fits the
Yeah.
same specs, you
Yes.
should be good.
Mm.
Yeah.
Doc, now, should we address the questions we had received before this seminar? We had
Yes.
like three
Yes.
questions,
Yeah, go ahead and stress that. Yes.
Yeah, I'm gonna post it, okay, one by one,
Mm.
so everyone can see the questions.
Yeah.
Um,
I think there's another question that came in too, real
Mm.
quick.
oh,
So
okay, yeah.
for
If so,
IND
yeah, let's address
submission
that first.
is a PK studies of intact ADC. I mean, generally for your you know, GLP tox and for your GLP tox, you will have all of your assays for looking at, total antibody, total ADC. You might have an assay for your free payload detection. So that would be certainly things that you would need validated methods for those for the assay. So yeah, I mean that's basically standard. Uh, it looks like there's another question. Let's see. Did you? Yeah, so I can answer this from the company, and then I think there's oh yeah, you posted. Oh, these are your questions,
Yeah, I
Kyoji,
just
but
posted the previous
the Q
questions.
and A
Yeah,
here.
yeah.
Um, for us, at least within Crossbridge, we've looked at a variety of expression levels, mostly around TROP2, since that's our lead. Although many of these cell lines express multiple tumor antigens, right? And so in our case, we have seen activity across a broad spectrum of TROP2 levels. I mean, typically from medium to high. We've seen activity, right? And so I think for us, if you think ahead to biomarkers, and especially for TROP2, there's differing views on is there an assay, and if it seems like you might be in the pharma space, and maybe it's somebody I know, I don't know, but obviously AstraZeneca has tried to come up with a biomarker for TROP2, looking at the cytosolic versus receptor. Yeah, I forget exactly what it is, but continuous scoring something is their thing. But by and large, I mean, it's going to be really interesting as the linkers get better. If expression level is going to matter. I mean, so far, with the exception of like HER2 low, and then that's parsed out. What does HER2 low actually mean? It hasn't fully correlated with expression level. I think, or linearly correlated, maybe is the way to phrase that. So I think it's TBD. And at least in our case, we're not planning to sort of stratify, but we're obviously, if you're working at, say, MD Anderson, you'll have the ability to get some of that data to, look by IHC and other methods that they normally do to get a sense of, is there a particular patient segment that you could enrich for that's going to have better activity. So
Yeah, I agree with Michael's opinion. The expression level is not linearly correlated with actual response because there are TROP2 or HER2 high cells that have also, high expression of, efflux pumps, or proteins, right? So in that case, even though the internalization rate is relatively high, the drug itself doesn't work well because of the, efflux mechanism. So it's, yeah, it's not, the simple, correlationship. Also, the degree of heterogeneity is also, an important, factor, determining the, overall efficacy. So in that sense, it's more and more important to test whatever new drugs in more, clinically relevant models, such as patient-derived, xenografts or PDX models, instead of trying to make perfect panel using the established cell line.
Mhm. Oh, yeah, there's another question. I think this one's you can answer, Kyoji.
GC versus UPC, which one would be better for homogeneous ADC? Uh, well, I don't know if this question is about the conjugation or the homogeneity of the resulting ADCs, but yeah, when it comes to the construction process, there's no difference in terms of EG-sit, between EG-sit and EB-sit. We can achieve almost, quantitative, homogeneous dark conjugation. And oh, the question is gone. But
Yeah,
well, yeah, let me summarize it. Yeah, so
no.
it's there's no virtual, substantial difference between those two linkers. What matters is, which, is better suited for your applications. In most cases, cytotoxic ADCs always, entail neutropenia. And in that case, you want to maximize the safety profile. That means, you want to use, the most stable EG-sit linker. But we also know that EB-sit cleavage kinetics is a little faster than EG-sit. And if your priority is to release your payload of interest, as soon as possible, and the hydrophobicity is not a problem, then EB-sit may be a better option. So this way, we can, fine tune the payload kinetics, profiles based on your need. So again, both linkers are great. They don't, give, really disappointing heterogeneous mixtures in any situations.
Mhm. Mhm. Let's see. Well, if there's not a question, honestly, maybe I think it'd be, I'm just kind of sitting here thinking in my head, we've got some really great scientists on the forefront of, the next wave of therapeutics. I'd be very curious maybe to hear from Kyoji and Jishang, like what are the modalities or the concepts that as you sit like on the leading edge are like things to be look out for or areas of unmet need or areas of exploration, things that are underexplored but has a lot of potential within, y'all's what you work on every day. I mean, I think that'd be really useful to hear
Yeah,
myself, honestly.
yeah. I think for the ADC, so you already working dual payload targeting TROP2 or other targets. So I think one of the directions today is actually bispecific antibodies. So antibody targeting multiple targets. On top of that, you have multiple payloads. So the molecule getting more and more complex. And so again, it's to play the synergistic effect of combination therapy. And these drugs will, of course, increase efficacy, but more importantly, to provide resistance. And that's where you already say just by simply doubling the payload by complementary mechanism cell killing, you can avoid resistance, single drug resistance. And I think the beyond uh, 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 drug conjugation. So Kyoji already alluded that antibody-drug conjugate can be including different payload like oligonucleotides, and so that's peptides. And this all oligonucleotide and drug combination. So just you can imagine all these modalities, and also antibody-drug conjugates actually are now moving beyond oncology indications for other diseases like neurodegenerative diseases, which is also a major direction in the future.
Very nice.
So yeah, payload innovation in the payload, not just structure but categories, as well as antibody innovation like bispecific or bispecific plus dual payloads or 2x2, could be a next wave. But again, the modality becomes more and more complicated, and we have to have a really strong rationale to justify, why we actually started that project. That's always a key, kind of challenge. Yeah, making such molecules is not really difficult using our platform, to be honest. But why you want to do that is more important. We should have, that homework done.
100%. And that's something that we at the company pride ourselves in, as we call ourselves a biology first company. And so any pairing that we work on, which is a lot more than what we've disclosed, we have I think an interesting rationale for why it would make sense to put two together. So yeah, I think if you stack that even on the target side, there's potentially some really interesting opportunities going forward. So
Absolutely.
Okay, good. So I do not see any more questions, and so we want to thank Kyoji and Marco for really cover a really important topic, not only academically but more importantly translationally. And so we wish Marco success, continued success for his cross bridge.